Okay, welcome to our chapter on DNA. And DNA is really a foundational molecule that we've just briefly talked about at the beginning of the course, but it really makes up so many of the different characteristics that organisms have, and it plays into all the rest of the chapters that we're going to learn about this year. So we're going to spend this week talking about the basics of DNA, and then the next couple of weeks talking about genetics and how DNA affects our bodies and organisms. So this is a really important week. Okay, you probably know a little bit about DNA already. I'm sure that you've heard it mentioned on the news or in scientific articles. It's something that really affects all of biology at this point, but it's pretty recent as far as discoveries go. We only started to learn about DNA and learn what we know now since about the 1940s. So we are going to go through some of the experiments and the scientists that have done those experiments that led to the discoveries of DNA, its functions, and its structure. Okay, the first scientist that we're going to talk about is Frederick Griffith. Frederick Griffith was a British scientist that was studying how bacteria make people sick. So the discoveries that he made about DNA were really accidental. They were a secondary conclusion because he was trying to study bacteria. Specifically, he was using pneumococcus bacteria, which is the bacteria that causes pneumonia. And he was working with two slightly different strains of pneumococcus. There were smooth colonies, and this was the virulent or disease-causing strain. It grew into smooth colonies on a petri dish. When he spread the bacteria on the petri dish, it actually grew into smooth colonies of bacteria. So that is the strain that caused pneumonia. And then there was a rough strain. That was the non-virulent or didn't cause disease strain. It's a harmless strain, and it grew into rough colonies. So it was easy to tell the difference, which was what was helpful for this experiment. He tried several experiments using combinations of these strains to determine how they were causing disease. He was trying to figure out how the bacterial disease was spread. His experiments involved injecting this bacteria into mice, and if the mice died, they had been infected with the pneumococcus bacteria. So he ran a couple of experiments. The first one is pretty obvious. When you inject a mouse with the virulent smooth strain, which is the disease-causing strain, when you injected a mouse with that, it died of pneumonia, which was to be expected. And then again, to be expected, when you injected a mouse with the non-virulent rough strain, the harmless strain, the mouse was able to live because that was not a disease-causing form of the bacteria. Then he tested the virulent smooth strain, the disease-causing strain, and he heated it. And when he heated that disease strain and injected a mouse with it, the mouse was able to live because the heat had killed that disease-causing bacteria, so it did not affect the mouse. Now, the really interesting part came when he added the heat-killed smooth strain from the last experiment. So he's taking this heat-killed disease-causing bacteria, and he's mixing it with the live rough strain. So this one did not cause disease on its own, and this one did not cause disease on its own. Both of those mice lived. But when he combined them together and injected into a mouse, the mouse died, which was a very unexpected result. So then he had to go about figuring out what caused that mouse to die. His conclusion was that both the heat-killed smooth strain and the rough strain were non-virulent on their own. Just as we mentioned, they did not kill mice on their own. However, when they were mixed, they did kill the mice. So this demonstrated that something, he didn't know what yet, was moving from the smooth strain into the live rough strain. Griffith suspected that it was a poison. So the poison, or whatever was moving from that heat-killed strain, was moving into the live, harmless strain, and therefore making it harmful. He called this movement of materials transformation. Scientists later realized that what was moving from the smooth strain to the rough strain was DNA, because bacteria, as we know now, but he didn't know then, transferred DNA back and forth all the time. Okay, so on to our next scientist. Oswald Avery set out to repeat Griffith's experiments, but this time he went a little bit further. So as you know, in science, we're all about repeating experiments and making sure that the results that other people have come to, the conclusions that other people have come to, are valid, and that those experiments are repeatable. Because if they're not, the conclusions are not valid. Okay, so Avery went a little bit further here than Griffith, and he used enzymes to systematically destroy molecules within the bacterial cells. So you used the same bacterial cells that Griffith did, but this time he destroyed different molecules systematically so he could figure out which molecules were actually moving from the bacteria, one bacteria to another. So first he killed the proteins, which was what scientists in that day originally thought was the genetic material, because proteins do so many things in your body. They suspected that proteins were what was causing all of the genetic traits and all the functions of cells. But he killed the proteins, and the results from Griffith's experiments were still the same. So that thing that was moving was still moving and killing the mice. So it was definitely not proteins. Then he destroyed carbohydrates. Then he destroyed lipids. Then he destroyed RNA. And finally, when he destroyed DNA, that was when the results of the experiments changed. Transformation within the bacteria occurred just as it had in Griffith's experiments. Only when the DNA molecule was destroyed was transformation unable to occur. So that was when Avery and other scientists said, wow, you know what? It's the DNA that's being moved from the heat-killed harmful strain into the live harmless strain, and that is what is changing the genetics of that bacterial cell. So it's really DNA that is causing the genetics of the bacteria. So the conclusion was DNA was the molecule being transferred from one bacterial cell to another, which killed the mice. Clearly, DNA was the molecule that carried the genetic instructions for the cell. And this was a radical discovery. Okay, then next we have a scientific team of Alfred Hershey and Martha Chase. And they, again, were looking to verify the conclusions that Griffith and Avery had come to. This time they went about it a different way. They used viruses for their experiment. And we haven't talked about viruses yet. We will, but this is what one type of virus looks like. This is called a bacteriophage, which is a virus that infects bacterial cells. Viruses are known to infect host cells by transferring a genetic component into the cell while leaving the rest of the virus outside the host cell. So there is some portion of this virus that is injected into a host cell, and that's what kills the host cell. They didn't know what molecule particularly was being injected from the virus into the host cell that was killing it. But whatever that was had to be the genetic material. So they needed to figure out what portion was being injected into the host cell and what was staying outside. Hershey and Chase wanted to determine whether it was DNA that the viruses were injecting into the host cell. If it was DNA, it would support the results of Avery and Griffith's experiments. So Hershey and Chase marked the inner portion of the virus and the outer portion of the virus with radioactive materials. So if we look back here, there's an inner portion here, and then there's this whole big outer portion. And they marked the inner portion of the virus and the outer portion differently. They used sulfur for the outer portion and phosphorus for the inner portion. So they're marking it radioactively, which is something that they can pick up on an X-ray. After allowing the viruses to infect the cells, they blended the cells and separated the viruses from the bacteria by centrifuge. A centrifuge is something that spins out, maybe you've seen it before, spins out different, it's a way to separate different solutions or different portions of a solution by spinning. Okay, so let's take a look at this here. Here are our bacteriophages, the viruses, and they marked the outside the first time with a sulfur label, radioactive label. It's like just a molecule that attaches itself. And the inside was injected. There was no sulfur inside the host cells at the very end of the experiment. So clearly that outside portion of the virus was being left outside. And they marked the DNA on the inside with a phosphorus radioactive label. And at the end of the experiment, the phosphorus was found to be inside the host cells, so they could tell that it was the DNA marked by the phosphorus that was being injected into the host cells and therefore killing the bacteria. So this is again evidence that DNA is the material that is able to be moved from cell to cell and affect the genetics of a cell. So the results clearly showed that the phosphorus labeled DNA had combined with the bacterial cells, while the sulfur-labeled proteins had remained separate from the bacterial cells. This demonstrated that it was the virus's DNA that was transferred into the bacterial cell, changing its genetic material. It was definite. DNA carried the genetic instructions. Okay, so now that scientists know that DNA is the genetic material, they needed to know what was the structure of that DNA molecule. They really had no idea. They had a couple of pieces of information, but they needed to work together to figure out what really does that structure of DNA look like. So scientists had determined that DNA, which stands for deoxyribonucleic acid, had to be a very special molecule. It had to be easily copied when new cells were formed, because as we talked about in mitosis and meiosis, every time a cell divides, chromosomes are copied, and those chromosomes are made of DNA, so it had to be easily copied. It had to correctly create an organism's characteristics, because all the traits of an organism they knew came from that genetic material, so somehow it had to correctly create all those traits. And it had to be accurately transferred from parents to offspring, because if it wasn't accurately transferred, then an offspring could look completely different from its parents, which they knew was not going to be the case. They did know that DNA had nucleotides in it. It's a string of nucleotides. They knew that. Each nucleotide is made of 3 parts. A 5-carbon sugar called deoxyribose, and that's partly where DNA gets its name, deoxyribonucleic acid. A nucleotide also has a phosphate group in it and a nitrogen base, and there are 4 different nitrogen bases: adenine, thymine, cytosine, and guanine, and you can see all the nitrogen in those molecules, and that's why they're called nitrogen bases. So here you can see a nucleotide right here. There's a phosphate group, there's a deoxyribose sugar, and there's a nitrogen base. Those 3 molecules together create one nucleotide. So scientists knew that DNA worked like letters in words. The sequence of the adenine, thymine, guanine, and cytosine created the information to code for all of a cell's genetic information. So the combination of the A's, T's, C's, and G's worked like letters in a word. That combination and sequence of bases is what created the unique characteristics of an organism. They just didn't know how that code was created or how it was transferred from one cell to another. Okay, so we're now moving on to scientists that have contributed to the structure of DNA, or discovery of the structure of DNA. Edwin Chargaff is the first scientist we'll talk about. He determined that the percentages of guanine and cytosine were nearly equal. So remember, there's 4 bases, A, T, C, G. And he determined that the G's and C's were almost always equal in any DNA strand, and that the A's and T's, or adenine and thymine, were also almost nearly equal to each other every single time. So that was a drastic discovery. They just didn't quite know what to do with it yet. Next came Rosalind Franklin, and she used X-ray diffraction photographs, which is a special type of photograph, to determine that DNA was helical and it was shaped like a spring. So she actually was able to photograph the molecule using X-rays to determine that it was shaped like a spring. It created an X-shaped photograph, and she could interpret that. So she came to realize that DNA was helical, and you can see that helical shape over here, that it's twisted. Next came Francis Crick and James Watson, and you may have heard of them. They are the most famous, by far, most famous scientists related to the discovery of DNA. They worked on models made of cardboard and wire for years to determine the DNA structure with no success. However, they caught a glimpse of Franklin's photograph of the X-ray photograph of DNA, and with that information they were able to determine the structure of DNA that we use today. So there's actually a lot of controversy surrounding this. There's a lot of people that feel that Rosalind Franklin did not willingly contribute this information to them, and that some information was stolen from her or without her knowledge, and she gets left out of the discovery, and Watson and Crick get all the fame and glory. So there's definitely some controversy about that. You can find lots of books and videos relating to that series of events. But regardless, Watson and Crick did come up with the model, and in that model they determined that a double helix was the shape of DNA, with a backbone made of deoxyribose sugar and phosphates, with the nitrogen bonded in the middle, adenine bonding with thymine, and guanine bonding with cytosine. So this, the blue and the gray, is the sugar phosphate backbone in this double helix. And so you have your deoxyribose sugar and your phosphate alternating all the way down, and then the nitrogen bases are bonded across the middle. And A always bonds with T, and G always bonds with C, and that is why Chargaff's rule of the A's and the T's being equal and the C's and G's being equal worked out in their structure. That was one of the ways that they knew that it was correct. Okay, so now we know that DNA is shaped like a double helix based on the model that Watson and Crick gave us. A double helix looks like a twisted ladder. The sides of the ladder are composed of alternating deoxyribose sugar and phosphate groups, as I just mentioned, and this is commonly called the sugar-phosphate backbone. The rungs of the ladder are made of the nitrogen bases. And as I just said, A always bonds with T, adenine bonds with thymine, and cytosine bonds with guanine. And this is called the principle of base pairing. So you can see that the cytosine and guanine are always bonded here, and adenine and thymine are always bonded together. And this is one of the rules that will get you through the whole rest of this chapter. So pay attention. A bonds with T, C bonds with G. Okay, since DNA is an essential molecule for all cells, it is important to keep it intact within the cell. And we talked a little bit about this when we talked about chromosomes in mitosis and meiosis. As cells grow and divide, this DNA must be moved without breaking or altering it. And chromosomes solve this problem. So this diagram here is called a karyotype. You'll work more with this in a couple of chapters. We'll make a karyotype. The chromosomes from a human cell in this picture are spilled out and organized by pairs, one from each parent. So here is all of the spilled out chromosomes, and they have them marked. Scientists can mark them based on the genes that they have. And then this is what a karyotype looks like if their chromosomes are all organized by pairs. Remember, because you get one from your mother and one from your father. So again, let's take a look at how chromosomes are formed from DNA. We start with long strands of DNA called chromatin, and those contain the DNA sequence. You can see the double helix here, and the A's and T's and C's and G's bonded in the middle. And that long strand of chromatin of DNA wraps around the proteins called histones, these little ball-shaped proteins, to keep it organized. And then the long wrapped strands of chromatin continue to condense until an X-shaped chromosome is formed. The center of the chromosome where the two sides meet is called the centromere. Again, most of this is a review, but just as we continue to talk about chromosomes and genetic material, I want to make sure that we have this crystal clear. Okay, so let's move on to another process that DNA uses, and we're going to talk about several processes in this chapter, in the following chapters. But today let's talk about DNA replication. DNA replication is the process by which DNA makes a copy of itself. And this is the process that occurs in mitosis. When the chromosomes are copied, this is what's happening. The DNA is replicating. This process is called semi-conservative because half of the parent strand is used for each new copy. And this was another situation where scientists didn't quite know how this worked until somebody did an experiment on it. This scientific team of Messelson and Stahl, who were American scientists, determined this property of semi-conservative replication using N15, which is an isotope, a heavy isotope of nitrogen that is found in DNA molecules. So in other words, it's a marker. It's just like the radioactive markers that we talked about previously. This is a way for them to watch molecules through a process. So every time the DNA copied itself, the density of the DNA sample was reduced by half. So they started with a heavy, dense molecule. Originally, the density was high. It was a heavy molecule because they had marked all the nitrogen in those bases. And then, as the DNA copied itself, they noticed that more and more of the bases did not have that marker in it. And that's how they could tell that the N15, that heavy isotope, was being reduced by half every time. The density was being reduced by half every time. So just like we say, I'm 25% Italian, if you have grandparents that are Italian, the genes are diluted every time they copy themselves. This constant dilution of the N15 showed that the DNA strand was dividing in half every time, each half being used to make a new copy. So I actually have a link here, and I will put that in in your lesson for today, but it doesn't work very well if I'm recording while I click on the link. So if you're watching this video, you'll want to go back and check on that link after you're finished watching this. So here's what this picture looks like. The video also does a really good job of explaining it. But this is what semi-conservative replication is. We start with the original strand, which we call the parent strand. And in DNA replication, the molecule, the DNA double helix, unwinds, untwists, and then unzips. And so you get half of the bases over here, one strand and then another strand here as it splits. And then two new strands are formed from the two sides of the parent strand. So because we have that constant base pairing rule where A always bonds with T and C always bonds with G, if we have half of the DNA strand, the other half is going to be automatically correct because it can only bond and pair one way. So we can create a whole nother strand from just that one strand. So here you can see that it is conserving the parent strand, but only slightly, and that's why it's called semi-conservative replication. And then as we continue, this strand is going to separate again, and then you have four, and this one will separate again, then you have four new strands. Two of them are going to have completely new strands in them that don't have any of the original parent strand, and then two of them are going to have half parent strands as that continue. That process is just going to continue on and on and on. But you can see how the Michelson-Stahl experiment showed that the original strand that had that really heavy isotope in it was getting diluted out every time, because then by the end of this experiment here you have just a few of those bases that are marked, and then you have a lot of the unmarked new bases. So that's what they were showing. So the blue strands are the original strands, and the green are newly formed, which we call daughter strands, just like parent cells and daughter cells in mitosis. You can see how the original strands, like those containing the N15, eventually became a smaller portion of the DNA set as more and more replications occur. And again, I know this is a slightly difficult concept, but if you click on that link and watch the video, that should help as well. So let's talk about how DNA is actually copied and how it's replicated. The nature of DNA itself makes DNA replication work. If A only bonds with T and C only bonds with G only bonds with C, a new strand can quickly and accurately be formed from any DNA sequence. So we just mentioned that. But you can see as this splits, the A here is always going to bond with a T, the C is always going to bond with a G, and then you will get an exact match of what was over here. And same thing for this side. So for example, if one side of the DNA strand has the sequence ACA, CTGA down this side here, the other side of the DNA strand will naturally have the sequence TGT, GACT. All of those are going to be the same every time because that is the only way that the DNA bases can bond. Okay, so here are the steps of DNA replication. First, there's an enzyme. There are multiple enzymes involved here, and you can tell it's an enzyme because it ends with ASE. Anytime you see anything that ends with ACE, that is an enzyme. So we talked about enzymes earlier in the semester, and we talked about catalase, and this time we're talking about DNA helicase. So helicase untwists and unzips the two strands of the double helix. And then DNA polymerase joins individual nucleotides to the two parent strands. So it untwists and unzips, and the two strands separate, and then the individual nucleotides are brought in by DNA polymerase, and they join to the two original parent strands, creating the four, just like in the picture that I showed you a couple slides back. Then the DNA polymerase proofreads the sequence to check the accuracy, because if any of those bases are wrong, we're going to have major problems. If a strand is inaccurately copied, mutations or mistakes can develop within a cell, and we'll talk more about that as we get into genetics. But really, a cell cannot afford to have any mistakes in that DNA sequence, or really serious things can go on. So if you have your DNA double helix here, it's already untwisted. The helicase is separating the two strands, and then the DNA polymerase is going to come along here and add in your new bases. Okay, and we will stop there for today, and next chapter we are going to talk about the other processes that DNA uses in order to create all the traits in your body.